A very good day to Dr. Yap and fellow viewers. Today, my groupmate and I will be presenting on the potential of 3D models in anti-cancer screening. To start off, I, Kimberley, will be giving you an intro of our presentation. In the quest of successfully launching a new drug discovery in preclinical phase and development in clinical phase is crucial, especially in the field of anti-cancer drug screening. Researcher has been heavily involved in producing new drugs that bridges the gap between experimental research and clinical trial. Recent advancement in bioprinting has produced several in vitro models such as 2D and 3D models that are widely used in drug screening. 2D models are traditionally used by researchers to evaluate effectiveness of anti-cancer drug towards cancer cell line. Additionally, it is to understand the molecular pathway of cell proliferation. 2D models are also grown in either shake flask or petri dish with grow medium as their source of nutrient. Meanwhile, 3D models are used by researchers to mimic tumor microenvironment and visualize the structure of cells. This presentation will be revealing the difference between these two models, the recent preference of researchers for 3D models, and case study that highlights the new direction for the future 3D bioprint model. I will now pass it to Zhi Yi.
Thank you, Kimberley. And now I will be talking about the differences between 2D and 3D models. First, the cells in 2D model grow in monolayer on flat surface culture media such as petri dish and culture flask, while the cells in 3D model grows in multiple layer in all directions. Besides, 2D model does not mimic tissue and tumor structure and also in vivo microenvironment, while 3D model does. The 2D model is lack of cell-cell or cell matrix interaction, while 3D model have. The cells in 2D model receive equal amount of nutrients from a medium during growth, whereas cells in 3D model does not. Furthermore, the 2D cell culture have higher cell proliferation rate, slower cell migration rate, higher drug sensitivity, and also lower cell viability, while the 3D cell culture has lower cell proliferation rate, faster cell migration rate, lower drug sensitivity, and higher cell viability. The gene expression in the 2D model is different from in vivo model, while the gene expression in 3D models is similar to in vivo model. 2D model requires lower cost maintenance compared to 3D models. And now I will pass the presentation to Ameerah.
Thank you, Zhi. I will be explaining why researchers prefer using 3D models opposed to 2D models. Firstly, 3D models would mimic the in vivo physiology of organisms, such as the histological architecture and heterogeneity. Also, they mimic native matrices and cell-cell interaction, as well as the interactions with the extracellular matrix. Another reason why researchers prefer 3D models are that 2D models are unnatural, while animal models are expensive and brings about ethical issues. Besides that, cancer cells cultured in 3D models reflect the behavior of cancer cells in their native in vivo environment. An example can be seen where cancer cells cultured in 3D models respond to chemotherapeutic treatments similarly to in vivo cancer cells. Now, I will be passing on the methods of 3D models generation for anti-cancer drug screening to Anjali.
Thank you, Ameerah. So first of all, before screening for anti-cancer drugs, we need to synthesize the 3D tumor models. But how to do so? There are actually various methods to produce 3D tumor spheroids. Based on a few case studies, the most common methods are hanging drop method, the force floating method, using low addition microplates, magnetic levitation, agitation-based methods, and microfluidic cultures. In each method, the spheroids are cultured in anti-cancer drug, and after incubation, they are later analyzed to screen for the most efficient drug. On the next slide, we have other methods of generating 3D tumor models using 3D extracellular matrices or ECMs and organoids. For 3D ECMs, we can either synthesize the matrices using natural polymers, non-natural polymers, or using biological sources. Once the matrices are made, the cancer cells are seeded and cultured in or on the matrix in the presence of a potential cancer drug. As for organoids, they can be stem cell derived or cancer patient derived. These cells are propagated in mice models, and different anti-cancer drugs are administered to find the effects of each drug, thus screening for the most effective drug. To screen for anti-cancer drug, in 3D models, the most common techniques include assays by luminescence, colorimetry, fluorometry, but there are also other methods such as flow cytometry, Western blot, Q real-time PCR, optical microscopy, electron microscopy, and 384 hanging drop array. Next, I'll be passing on the slides to Anis, who will be going through a case study about 3D bioprinted cancer models used in anti-cancer drug screening.
Thank you, Anjali. The case study I'll be reviewing is on 3D bioprinted cancer models to test anti-cancer drugs. An NBIB-funded research conducted recently by a team of researchers from the University of Minnesota has developed a newly dynamic and efficient 3D bioprinted tumor model for anti-cancer drug screening. The 3D model was made in a laboratory dish and functions in studying the primary site, growth, and spread of cancer tumors in the body, thus tackling the recurring problems in which previous 2D models could not replicate the conditions and outcomes of tumor growth in the human body. The 3D bioprinting technology used in the project originated from UMN lab through Michael McAlpine. Through this research, 3D bioprinted biochemical capsules were combined with 3D bioprinted tumor cells. Through 3D bioprinting technology, melanoma cancer cells, lung cancer cells, normal cells, and blood vessel-like structures are able to be precisely located in the laboratory dish based on their individual functions. Moving on to the next slide, the figure on the right shows a schematic of the case study's in vitro tumor model. It demonstrates the integration of tumor cells, the blood vessel-like structures, and chemical gradients. Chemicals that guide cancer cells migration or the growth of blood vessels are a swell tag in the cause of hydrogels. They are also encapsulated within an outer shell made of gold nanorods. A time-controlled release of the capsules are activated by laser light, which then creates a chemical gradient that ultimately guides targeted cell growth. Thus, these features provide a 4D control over both space and time. McAlpine further emphasizes that the cells and capsules are precisely printed in biologically relevant sites, and the chemical depots propel movement upon a triggered release. This is a dynamic 3D tissue engineering system, giving the user control over the diffusion process at some later point after the printing process. The researchers have a system that can collect cells that have self-selected to enter blood vessel and become metastatic. They believe that their scalable model may help to further understand the spreading of cancer cells and formulate even more effective treatments. To summarize, with cancer being one of the leading causes of death, there is an urgent need in drug research at the preclinical level to develop novel molecules to potentially manage these incurable diseases. Technological investment in studying the mechanisms of cancer progression and prognosis have been progressively made with the development of 3D models. 3D spheroid-based models especially being a promising tool in generating high-throughput screening platforms and inexpensive in vitro metastatic models. However, through current technologies, we still have a long way to go in creating a fully recapitulated tumor microenvironment in vitro. This is due to the simplicity of spheroid models compared to clinical tumors, we should alert researchers to not overestimate the accuracy in predicting potential response to drugs. With that, I end our presentation. Thank you for watching.